Then we move to the third speaker. The third speaker is today is Dr. Sunjae Lee. He actually is been friend of me since 2014. We met back then when I was still in Sweden. So let me briefly, Dr. Sunjae. Okay, you're here. Yeah, sorry. Good. So let me introduce your CV quickly before we begin your talk. Dr. Sunjae Lee completed his PhD in 2015 from the Kite University. I believe this is one of the well-known research institutions worldwide, because I know so many great professors from there. And then 2014 and 15, he went to the Sweden to feel the cold climate. And then he did a postdoc there in the group of system and synthetic biology, where later on from 2015 to 18, he did the second postdoc in the KTH, where it was in Stockholm. It's a Royal Institute of Technology in Sweden. And then from 2018 and 2020, he then moved to the UK, to the King College, where he started to work in the translational system biology group. I think he was looking at the interaction between the host and microbe interaction. So maybe in short word is that microbiome study. Yeah. And then I think recently he's just feel maybe tired of the European culture. So then he moved back to Korea a few months ago and established his own lab in Korea in the University of, in the School of Life Science, Gangju Institute of Science and Technology. So we are very honored to have you, Dr. Sunjae Lee, for the talk today, which is going to be in the title of the System Biology of Metabolism for the Understanding of Human Physiology and Microbiome. So without further ado, please join me to welcome Dr. Sunjae for his interesting talk. Welcome.
Thank you. Thank you for your nice introduction. So I will share my screen first. Can you see my screen?
Yeah, very well.
I'm really thankful for the nice opportunity to present my work. And I'm really happy to interact with many researchers in this metabolomics field. I'm not good at metabolomics itself, but I'm very big fan of utilizing metabolomic data to understand human physiology. So today I want to talk about the systems biology of metabolism for the understanding of human physiology and the microbiome. I think let me check. I'm sorry. Yes. So I think the people here surely knows how metabolism is so complex, right? So there are many challenges in understanding human physiology. The first, as you can see in this map, this metabolic network is so complex. So everything in this human physiology is very complex, and then it does not work just by alone. They work together, so they are all interconnected, then make more complex. And there is another obstacle, which is dynamic behavior. So we hope that this network, this system is stable and does not change, but actually by time it changes a lot. That's why such kind of complex characteristics make us too difficult to understand human physiology. So people, they try to do their best to understand human physiology. So they develop two kind of aspects. First of all, we need a good map that can explain systems level. And then we do observe many times as possible. So this based on data-driven understanding, we can simplify this complex human physiology into very simple biological clinical readout. For understanding, data-driven understanding, sometimes we develop the metabolic model for the system's understanding. And also we try to measure this complex system, for example, transcriptome, metabolome, and metagenome. And based on those very beautiful map and data, we can easily understand the human physiology. So I want to give you one example study of this systems biology understanding. So I think everyone knows that low carbohydrate diet is very good for our health, right? So there is an interesting study of a low carbohydrate intervention study to fatty liver patients. So the conclusion of this study is simple. If you have this low carbohydrate diet, isocaloric, then you will see that your liver fat decrease in just in 2 weeks times, and you will also observe that inflammatory marker also drop down. So we know that this low carbohydrate is very good for your health. But another question is that then how this low carbohydrate diet can reduce the liver fat and help our health. That's why in this study, they have a beautiful metabolic map, and also they measure different kinds of omics data, let's say multi-omics data. So based on microbiome metagenome data, we see that microbiome composition changed by time like this way. And then one of the notable feature here is that many kinds of short-chain fatty acid producing bacteria is decreasing. And also, as you can see here, that actually the total short-chain fatty acid is decreasing. So that means that energy harvested by microbiome is decreased by low carbohydrate diet. And also, based on liver transcriptome and genome scan model, we observed that glutathione metabolism is activated, and also we also see that folate synthesis by microbiome was increased. So in conclusion, they support the fat burning. That's why we see that low carbohydrate can decrease the liver fat. So likewise, if we have a good systems model and multi-omics data, we can understand how our human physiology is changing. So thankfully, I was able to participate in an interesting systems biology project. So today I want to introduce you this interesting systems biology project. So I think most of people here maybe know already about obesity can progress into insulin resistance and also type 2 diabetes. And also we know that blood glucose is a remarkable indicator of this disease progression. But in this study, we found that one of the minor sugar, mannose, actually can be associated with the disease progression, and even is more stronger indicator of this disease progression than blood glucose. So today I want to introduce this interesting story with you. Before explaining my project, I want to a little bit explain more this complex systems biology model. So the systems biology community work together and try to make good resource for the community, which is tissue-specific genome-scale metabolic models. So basically, if you have this model, you can simulate this model and you can predict metabolic behavior, and you can understand tissue-specific metabolism and also disease-wise dysregulation. In 2013, adipocyte model was developed, and 2014 hepatocyte, and 2015 muscle germ-scan model was developed. So based on this model, many people can understand, for example, muscle metabolism, diabetes, or some NAFLD patients, liver tissue metabolism. One of the major obstacles in this model, sadly, is that it does not have any information about cellular regulation, because genome scan model is a set of biochemical reaction. But what is lacking is that it does not have any transcription regulatory network or protein interaction network. So we are not sure how cell want to regulate this metabolism. That's why I try to integrate biological network to the model to understand this regulation of metabolism. So based on my network analysis, I can point out, oh, this metabolic part was dysregulated or not. So to understand this obesity, insulin resistance, and type 2 diabetes, I built a cell-specific biological network for the most interesting organ like liver, adipose tissue, and muscle. And based on this interesting network, I tried to identify dysregulated metabolism. So up to here, I know it's very complex, it's very difficult to understand. So I decided to do very simple analysis on this network. So which called coregulation analysis. In the network, for example, if we have enzyme 1, enzyme 2, we can find that there are some transcription factor binding together with the enzyme 1 and 2. If there is a 2 transcription factor binding together with the enzyme 1 and 2, then we can say that it has a higher probability to co-regulate it and also co-express. If we find 4 or 10 transcription factor binding together enzyme 1 and 2, then we simply think that this enzyme 1 and 2 is highly co-regulated, and cell systems think this is a very important part of metabolism. So I counted this co-regulation strengths, and I do some simple statistics, try to find what part of metabolism is highly co-regulated. So based on this methodology, I checked all the 3 different tissue types and tried to see what part of metabolism is highly co-regulated. So here, interestingly, we found that the fructose mannose metabolism, actually one of the highly co-regulated metabolism in all of the tissues. Of course, we can find that some tissue-specific co-regulations correlated to the part. For example, cholesterol biosynthesis is highly regulated in the liver tissue, and propionate metabolism in muscle, adipose tissue. Another interesting is this result is from the normal condition, but I generated a network, lean subject network and obese subject network, and compared the network property and found that fructose mannose metabolism has decreased co-regulation strength. That means that there is some dysregulation happening in this mannose metabolism. That's quite interesting. And then we do another independent analysis, which is called repertoire-metabolic analysis. This analysis simply the statistic checking the gene expression patterns, catalyzing some specific metabolites. For example, if enzyme catalyzing this metabolite increase, then we can imagine that this metabolite may produce more. But if enzyme expression here decrease, then we may expect that this metabolite may be decreased. So based on that kind of analysis, we can identify that the mannose uptake in liver tissue was significantly decreased. So if the mannose is now well consumed in the liver, that also means that plasma mannose can increase. And after finding this regulation, we try to understand why this mannose decreased and less used in liver tissue. So we try to check many literature to understand this mannose metabolism in insulin resistance. So first, if less mannose consumed in liver, because mannose is the major building block of the glycosylation. So if mannose is not well consumed in the liver, the hepatic glycoproteins will not work properly. And if one of the major glycoprotein in liver is actually insulin receptor. That's why if mannose is not well consumed in the liver, eventually affect the function of the liver insulin receptor, and eventually you can see that many free insulin in the blood will not bind to these insulin receptors, eventually lead to the insulin resistance. So it's quite interesting hypothesis. And we also found that this insulin receptor in liver is not small portion. Based on imaging paper, we found that actually more than 50% of the insulin actually binding to liver tissue in insulin receptors. So actually, liver tissue is very active organ for insulin clearance. So that's why if insulin receptor in liver have some problem, then it really leads to the insulin problem. And also we also found some the experiment paper. So if we inhibit the glycolation, then it really affect the insulin receptor function, and then insulin does not enter into the cytoplasm. So actually, if we block the glycosylation, it has a problem. And also, we actually also see that if we knock out the liver tissue insulin receptor, there are some studies show severe insulin resistance. So interestingly, there are many evidences out there, but no one really collect together and make this story together. That's why no one really tried to understand this mannose metabolism in insulin resistance. So we did not stop here. We do further validation using metabolomics cohort. So we have a 3 metabolic cohort. The first cohort, we have a lean and obese subject. Another one, we divide obese patient into insulin sensitive type and resistant type. And the last one, we have a high insulin secretion versus low insulin secretion. So as you may expect, blood glucose level is different between lean and obese. But interestingly, mannose also quite different between lean and obese and correlate with the other clinical parameters like glucose. But when it comes to insulin sensitive versus insulin resistant obese patients, this mannose is very significantly different between 2 groups, but it only highly significantly correlated with the other clinical parameters. So it looks like mannose is more like driving factor rather than just effects of the glucose changes. And as you can see here, if we have a high insulin secretion and low insulin secretion group, we also observe the similar patterns. So less insulin secretion group has a highly enriched mannose and correlate with the other clinical parameters. So basically we found that less mannose uptake can lead to the insulin resistance, and based on the metabolic cohort, we observed that really mannose is increased in these insulin resistant patients. So that result has been highlighted in Nature Endocrinology, and our group decided to validate more in other metabolic disease as well. For example, cardiovascular disease, and then we found that this increased mannose not just observed in insulin resistant type 2 diabetes, they are observed in other metabolic diseases as well. And another interesting is, so up to here, we know the mannose less consumed in the liver tissue, it leads to insulin resistance. But then if we supplement the mannose, maybe it's really good for our health, right? So there was one group did something that we really want to do. So this group, they supplement the mannose to the high-fat diet mice and then see how phenotype changes. Interestingly, even though these mice have a high-fat diet, it has also mannose supplementation, then it does not increase the body weight. But this effect does not recapitulated by the galactose. And then monosupplementation can reduce the liver and adverse fat. And another thing is that actually this monosupplementation also changed our microbiome, and it leads to less energy harvest. That's why it leads to this kind of good phenotype. So here it's quite interesting. Human physiology is so complex, and not just one part is working alone. It is connected with a different part of the biological process, including microbiome. So today I also want to little bit introduce how our human microbiome shaped. Actually, human microbiome is also very complex, and also we need big data analysis because, as you might know, the human microbial cell, they in terms of the number, they are 10 times larger than human cell. In terms of gene numbers, they are 100 times larger than human genes. So they harbor the big number of cells and the genes. So they are quite difficult to understand. So we need some kind of big data analysis. Up to now, microbiome is known to affect host metabolism and immune development. So if you take some fiber, the microbiome have to break down this resistant fiber and synthesize good metabolite to us. Another interesting is that it also have immune development. So if you have a germ-free mice, basically many people say that, oh, germ-free mice is basically sick mice because they don't have any immune system, because they could not develop immune system by microbiome. So the microbiome is very important for two aspects. So we try to better characterize this human microbiome, this complex human physiology, using big data analysis. So here in this project, I tried to define what is the health microbiome and disease microbiome. So luckily, we can find around 5,000 samples, public sample up to now, from the 80 countries and 28 disease cohort, and try to understand this human microbiome. The first, I want to show how our global health microbiome looks like. Based on microbiome composition, so here is each subject from the different country, and they are all healthy condition. When you plot the microbiome composition of different subjects, we can see some clear distinct cluster here. So here is a European cluster, and here is a kind of rural area or non-Western countries. And here, so here I'm not sure why they are clustered separately, but they are Chinese, Japanese, and US population. So they are all separate into three big different clusters. This cluster is not just based on the composition. When it comes to the microbiome function, we can find some different enrichment of the function. For example, some industrial countries like European and US, we see that antibiotic-resistant genes are more enriched in those country species. We can also find there's some mucin-degrading enzyme, which is really bad thing because it degrades the mucin in our bowel, and the bacteria can leak into our human portal vein. So that's kind of a bad thing. Also, we can observe the secondary metabolism as well. So we found that microbiome, our global microbiome, also very different by geographical cluster. And we also try to associate microbiome changes with the clinical parameter as well. So based on the 1-year longitudinal cohort, we tried to associate the changes of the microbiome and clinical parameter, and we found them actually many kinds of clinical parameters associated with the microbiome changes. One of the most significant association was this Bacteroides fragilis, which is immune modulating bacteria, which was highly correlated with the CRP level and troponin T level, which is inflammation marker and heart failure marker. So maybe those changes really occurred by the Bacteroides fragilis. And this kind of association study is not just association study. Actually, there are many experiments approved that verify that it really microbiome drive the clinical parameter changes like weight and liver fat and blood glucose. So up to here, we defined how our health microbiome defined. But now I want to define disease microbiome. So I did this analysis from the 28 cohort from 11 countries. So basically we know that which bacteria is enriched in which cancer, which geographic region. And then after do this pan disease analysis, I summarize the simple message, what is the disease microbiome. So based on this plot, we can find that commonly enriched species in disease here, and commonly depleted species in disease. And these species, commonly enriched species, have a characteristic like some oral origin species. Our data is the gut metagenome, but interestingly, some oral origin bacteria start to populate in our gut in disease condition. And also we can find then some species living in low diversity. So they are kind of bed bugs occupying all the resources in low diversity. So it's kind of weed. So they are kind of enriched in disease condition. And when I look at commonly depleted species, I can find that one of the well-known good bug, Akkermansia muciniphylla, and also many kind of commensal bacteria. And another thing, we found that some bacteria highly enriched in that geographical region, they are decreasing in disease context. So basically they lose the environment contact, and it's a bad thing for your health. And we try to find the functional cluster rather than just single function annotation to understand our microbiome functional changes by disease or health. First of all, when I find the functional cluster, we found two big, large function cluster. So here, the x-axis is number of species having this function, and the y-axis is cluster size. So here is basically the functions very highly preserved in the many kind of commensal bacteria. And this function include the glycolysis and biosynthesis of amino acid and folate metabolism. So basically, this kind of function is actually the major role of the health microbiome. So it's kind of expected, but we also observed another big cluster we call the pathocluster. So actually, this cluster have some kind of bad function for the host. So basically, they do hijacking nutrients. So they rob nutrient from the host, and also they produce a CD4. They do iron uptake from the host. So this function do the bad thing for the human health. And then after having this kind of function cluster, we see which function cluster enriched in disease or depleted in disease. And then we found that normally nutrient uptake, those kind of ascorbate PTS system or Manos PTS system, those kind of nutrient uptake, that kind of function is enriched in disease. And also we observed many kind of mobile element enriched on oxidative damage related functions are enriched. So basically these functions are key signature of our disease microbiome. Yes. So thank you everyone for today's talk, and nice to present my work with you. And please have any question to me. Thank you.
Okay, thank you very much, Dr. Sunjae. So so we have a time for few questions. So any questions from the audience? Okay. There's a one question from the audience. Okay, can you see that, Sunjae?
Yes, question, is it possible to use information from co-regulation in flux analysis? I think it's kind of good question to me. So actually, what I want to do in the final goal is to somehow combining this flux analysis together with our co-regulation analysis. So up to now, this method does not apply to flux analysis because we just check the gene expression pattern. But in the end, if we can combine co-regulation with the flux, then we can predict what is the problem in this metabolic regulation. So yeah, that's a good question.
Okay, maybe I have another question. Maybe just from the beginning. So if you have a model which is working well with obesity, but if we wanted to study other diseases, can we have a scaffold model where we can take the model from the obesity and then readjusting, or we have any scaffold model where we can use as a scaffold and then start to build up on that?
Yes. So the one good thing of this network and model is that we can apply to different types of disease, not just for obesity and insulin resistance. So because this model is quite a generic model, so only thing we need is just we need the RNA-seq from the patient. Without this, we are not sure which part of metabolism is dysregulated. But other than that, it's applicable to any types of disease.
Okay, thank you. There's one more question from the audience, I think from Dr. Kwanta. Yeah.
So I was wondering, how can you normalize metagenomic microbiome data obtained from different 28 cohorts? Yes, so this one was quite good question. So I think in the metagenome part, the normalization one of the issue. So first of all, I used the same reference gene catalog, and then we mapped all the metagenomic data to the same reference. And then based on the sequencing depths, we rarefy them into same level of depth. That's why we are kind of do our best to normalize our metagenomic data. But of course, some of the data, for example, because reference data does not have some non-Western country, sadly. That's why it does not applicable to many other non-West country. But other than that, it's quite well normalized and applicable to many different types of data. And then there is another question from Jutamat. Do you have information on alteration of microbiome profiles at fibrin level? Changes short chain fatty acids leading to support fat burning, slide 6. Okay, so that study, so actually, I think the definition of short chain fatty acids. So we know that there are many microbial species can produce short chain fatty acid, but it's a little bit difficult to really point out what species are really producing these short-chain fatty acid. And also it depends on the microbial interaction. Sometimes it produces something, but it depends on the other species to produce that short-chain fatty acids. So it's a very complex thing. So in the study, based on just previous known species information, we just took the microbial species and checked their population changes. And eventually we just directly measure the short-chain fatty acid concentration. So that's why we could conclude that the decreased short-chain fatty acid help us. But then, yeah, it's a little bit difficult to have a microbial profile really associated with the fat burning directly. Yes.
Okay, thank you very much, Sunjae. I think we a little bit over time, so maybe we just we still have a panel discussions afterward, so we can discuss more in detail of some specific points that anyone concerned. So I think I would like to invite everyone of us to join me to thank for the great talk from Dr. Sunjae again, and then we move on to the next speaker, please.